Hey everyone, welcome back to my podcast, Anatomy and Physiology Bit by Bit. This is your host, Dr. Steve Sullivan, once again from Bucks County, Pennsylvania, which is just outside of Philadelphia in the United States. Well, today we're going to finish up the immune system by talking about adaptive immunity. Adaptive immunity is the third line of defense against pathogens and diseases that our bodies have. So we're going to get pretty deep into that. I do want to discuss something real quick because after we're done with immunity, we move into the digestive system. After the digestive system, we're at reproductive systems, and then we'll talk a little bit about maybe human development and some genetics. And then we are through a typical 2-semester anatomy and physiology for majors course. So I've been thinking a lot about what's next? What is next for this podcast when we finish the course? And I had a pretty good idea what I was going to do. But then I got an email from Ryan, who is a first-year medical student in the United Kingdom. Hey, Ryan, thanks for emailing me. And his suggestion was exactly what I was thinking. He suggested that I spend time every episode on some of the most common medical conditions that exist in humans. And that is what I was going to do. So that is my plan. My plan is that this podcast will pivot to being about pathology and about diseases and conditions and injuries that affect the body systems that we study in anatomy and physiology, especially when it comes to maybe some very interesting rare diseases, but especially when it comes to some of the things that, as a clinician, you're going to see most often. I think that's the best direction that we can go. We're a little ways off from that. I don't know how many episodes it's going to take to finish up this course, but eventually, I think that's the direction we're going to go. So thank you, Ryan, for reaching out. You hit the nail on the head. We were thinking the same thing, and I think that is the best way to go. All right, so we got a lot to talk about in terms of adaptive immunity, so we're going to get right into it. Adaptive immunity has 3 main features that set it apart from innate immunity. 1, there's a systemic effect. When the immune system responds to a specific threat, like a bacterial or viral infection, it works throughout the entire body to eliminate that pathogen, no matter where it is. It doesn't matter. Innate immunity typically is local. We talked about that. It also demonstrates specificity. Adaptive immunity targets a particular pathogen. Compare that to innate immunity, which we covered in the last couple of episodes, which is nonspecific. It targets any pathogen. The specificity of adaptive immunity means the response to one pathogen, like the flu virus, for example, won't be effective against a different pathogen, like, say, the chickenpox virus. The immune system can respond to billions of different pathogens. That's billions, with a B. That is a lot of pathogens that we can become exposed to. And finally, adaptive immunity has memory. Once the immune system has dealt with a pathogen, it remembers it. So if the body encounters the same pathogen again, the immune system can respond so quickly that you might not even feel sick. With innate immunity, the response time is just as long for the second exposure as it was for the first. Adaptive immunity is split into 2 types, cellular immunity and humoral immunity. Even though they're different, these 2 types often work together to fight off the same pathogen. Cellular immunity, which we also call cell-mediated immunity, so you might see that in some of your sources as well, uses lymphocytes that directly attack and destroy foreign cells or infected cells in your body. It's especially important for fighting off pathogens that hide inside your cells, like viruses, bacteria, yeasts, and protozoans. It also targets parasympathetic worms, cancer cells, and transplanted tissue cells. Humoral immunity uses antibodies, so it's also known as antibody-mediated immunity. While antibodies don't directly destroy pathogens, they mark them so other parts of the immune system can take care of their removal. The term humoral comes from the ancient Greek word for body fluids. Since antibodies are found in the blood plasma, lymph, mucus, saliva, and breast milk, we call it humoral immunity because those are some of the body's humors. Humoral immunity is most effective against bacteria, yeasts, protozoans, toxins, allergens, and viruses that are still outside of cells. When microorganisms invade host cells, they're usually hidden from attack by antibodies, which mostly work on extracellular stages. However, cellular immunity can still target and destroy these infected cells. In some cases, humoral immunity and cellular immunity can even work together on the same pathogen at different points in its life cycle. So let's talk about the different classifications of immunity, because we classify them into active or passive immunity and natural or artificial immunity. Those are the categories of immunity. First, we'll define active and passive. Active means your body's producing the antibodies or T cells. Passive means your body is given antibodies, already produced somewhere else. Remember, active means you're doing the work. Passive means you are being given something or someone else is doing the work for you. Natural and artificial refer to whether the exposure to the pathogen happened on its own in the environment or if it was medically introduced on purpose. So we'll talk about ways to apply these. The first example is natural active immunity. This happens when the body makes its own antibodies or T cells after being exposed to a pathogen like a virus or a bacterium. And that exposure happens in the environment. For example, your friend has the flu, you get too close, and now the flu virus is in your body. Your reaction to that flu virus that protects you from that same virus next time is natural active immunity. It happened in the natural environment, and you are actively going to make those antibodies. Then we have artificial active immunity. This is when your body produces its own antibodies or T cells, it's active, after being vaccinated against a disease like tetanus, chickenpox, or hepatitis B. Vaccines contain weakened or inactivated pathogens that are present enough to stimulate an immune response, but too weak to cause illness. Some vaccines also require booster shots to keep the immune system's memory sharp so we continue to produce the antibodies in protective quantities. Some viruses mutate rapidly as they replicate, changing their genetic makeup and therefore their antigens. So, for example, a new flu or COVID vaccine is required each year to keep up with the latest version of the virus. So this is artificial active immunity. It's active because you're still producing the antibodies or T cells against the pathogen you've become exposed to. But it's artificial because it was introduced in a medical setting. It didn't just happen in the environment. A weakened or inactivated pathogen is called an attenuated pathogen, and that's what's used in vaccines. It's not enough to actually make you sick. You're not going to get the flu from the flu vaccine. But you still may make antibodies against it. So when you become exposed to that flu virus in the natural environment, you already have the defenses against it and you don't get sick. Or you get a lot less sick than you would have otherwise because you have a big head start. The third category is natural passive immunity. This is when a person acquires antibodies from another person, like a baby getting antibodies from the mother during pregnancy or breastfeeding. So antibodies that the mother's already made is being passed on to the baby who is nursing or the fetus who is still inside the uterus. So it's natural because it's not being introduced in a medical setting, but it's passive because the fetus or infant isn't the one producing the antibodies. Finally, artificial passive immunity. This is when antibodies are obtained from another person or animal and then injected into the body in a medical setting. This is often used for emergency treatments, like you get bitten by a snake, and now there's snake venom coursing through your blood, and you need to destroy that venom, those toxins. And so since you don't have time to make your own antibodies, we give you antibodies to take care of that venom that's already produced. Another one would be botulism. Botulism is a toxin, or I should say botulism is caused by a toxin from the bacterium Clostridium botulinum and can make you really sick. The treatment requires giving you antibodies against that toxin. And then rabies is a real popular one. If you get bit by an animal that may be rabid, you don't have time to make antibodies yourself against that rabies virus. Rabies virus is the deadliest virus. So instead, you get rabies shots, and those rabies shots include the antibodies against the virus so that you can prevent the fatal illness that results from a rabies infection. Now, only active immunity, the one where you're producing the antibodies and T cells, provides long-term protection since it has memory. You're going to keep producing those antibodies and T cells. Whereas with passive immunity, it might only last for a few weeks before the antibodies are broken down by your own system, and then they're gone. So the active immunity is the one that has memory. So now let's talk about antigens. Any molecule that binds to an antibody is an antigen. Some antigens are free-floating, like snake venom or foodborne substances, while others are part of a cell or organism structure. For example, antigens are present in the cell wall of a bacterium and the plasma membrane of an erythrocyte, for example. That's where we get our blood type from. You can go back and listen to my podcast episode on blood type and learn about the antigens that make us type A, B, O, A, B, or A positive, A negative, etc. Viruses also have antigens, even though they're not organisms or cells. Generally, antigens are large and complex molecules that are unique to each organism, which allows the immune system to tell the difference between the body's own cells and foreign invaders. Not all molecules are antigenic. Small molecules, like glucose and amino acids, are harmless to the immune system because they don't have antigens, but larger molecules like proteins and polysaccharides can trigger an immune response. You might have heard the term epitope. Epitopes are specific parts of an antigen that the immune system recognizes, and each antigen can have multiple epitopes, leading to the immune system's production of different antibodies for the same antigen. Some smaller molecules, like haptens, can't trigger an immune response on their own, but they can when they bind to larger molecules in the body. What they do is they create a new complex that the immune system sees as foreign. Many allergies, like those to cosmetics, detergents, or poison ivy, are caused by haptens. In severe cases, like with penicillin or a peanut allergy, the immune system can react so strongly that it causes a severe, life-threatening reaction called anaphylaxis. In anaphylaxis, the immune system releases a flood of chemicals that can cause the body to go into shock. There is a quick and sharp drop in hydrostatic blood pressure, the airways of the lungs narrow, which impedes ventilation, and the heart rate may become fast with weak contractions. There may also be an associated skin rash, nausea, and vomiting. Anaphylaxis needs to be treated right away with an injection of epinephrine, which counters these effects. That's why you see people who have these allergies carry EpiPens around with them, because they need that emergency shot of epinephrine should they get stung by a bee if they're allergic, or inadvertently consume a tree nut that they're allergic to, etc. You need that EpiPen to combat the anaphylaxis. Now let's talk about antibodies, which are also known as immunoglobulins. They're proteins that play multiple roles in our immune system's defenses. They come in different forms, and some are embedded in the membranes of immune cells, while others are free-floating in our body fluids. The structure of an antibody is made up of 4 polypeptide chains, with 2 heavy chains and 2 light chains. The 4 chains are held together by disulfide bonds forming the shape of the capital letter Y. The ends of the Y's upper extremities contain sites that vary in structure from one antibody to the next. Because of that, we call these the variable regions, or the V regions. They're variable because they are structured to bind to specific antigens when we become exposed to them. For this reason, the ends of the V regions are called antigen binding sites. The rest of the Y is called the constant region because it doesn't change. There are 5 types of antibodies: IgA, IgD, IgE, IgG, and IgM. The Ig is for immunoglobulin. Remember, antibodies are also known as immunoglobulins. Each class has different roles in immunity. For example, IgG is crucial for newborn immunity since it can cross the placenta from mother to fetus. And IgA is passed from mother to child through breast milk. The immune system can produce a vast variety of antibodies. Some estimates say it can make up to 1 trillion different ones. Trillion with a T. This diversity comes from a combination of processes like somatic recombination, which mixes and matches DNA segments to create unique antibody genes, and somatic hypermutation, where B lymphocytes make high rates of mutations to generate even more diversity. The diversity allows us to fight off a wide range of pathogens. It's pretty amazing how our immune system pulls it all together. All right, let's talk about T lymphocytes. If you listen to the episode on leukocytes, you'll recall that T lymphocytes, which we also call T cells, they're the same thing. They're produced in the red bone marrow by hematopoietic stem cells. And after they're created, immature T cells enter the bloodstream and migrate to the thymus, where they undergo critical development and 2 difficult tests that determine if they can become functional immune cells. Remember, the thymus is much larger when you're a fetus and when you're young than it is when you're an adult. It does degenerate over time. But it exists above your heart in your mediastinum. When the T cells get to the thymus, they head to the cortex and group around cortical epithelial cells. So that thymus, and you might remember this when I talked about lymphatic organs. So if you go to the episode on lymphatic organs, you'll hear me talk about the anatomy of the thymus. The thymus is split into lobules, and each lobule has a medulla, the middle portion, and a cortex, the outer portion. Just like so many other organs and structures in our bodies with a medulla and a cortex. All right, back to T cells. The T cells head to the cortex and group around cortical epithelial cells. These epithelial cells evaluate whether the young T cells can recognize foreign antigens and antigen-presenting cells, which we call APCs, by the way, and make sure that they can also recognize your own antigens. If a T cell has the correct receptors, the epithelial cells send a survival signal that spares the T cell. If a T cell fails to recognize antigens, it doesn't receive this signal. It can attempt to take the test again by rearranging its DNA to form new receptors. However, if it fails a second time, the cell dies via apoptosis within a few days and is cleared away by the cortical macrophages. T cells that succeed in recognizing antigens are called immunocompetent. This initial testing process in the cortex is called positive selection, and only a small fraction of T cells make it through. Those that pass move deeper into the thymic medulla for further testing. I'll post an image of the thymus on Instagram so you can take a look at the cortex and medulla and notice that the thymic medulla is less dense than the cortex. This is because so many T cells have been weeded out before reaching the medulla. In the medulla, those T cells that do make it through face a second, even tougher test, which is conducted by macrophages and reticular cells derived from bone marrow. Here, the goal is to eliminate T cells that mistakenly react to the body's own antigens, or antigen-presenting cells, without an antigen present. If those cells survived, which does happen sometimes, they could potentially cause autoimmune diseases. To prevent this, medullary cells expose T cells to self-antigens presented on major histocompatibility complex proteins. We call these MHC proteins, major histocompatibility complex. Autoimmune means the immune system attacks the self. Some examples of autoimmune diseases are lupus, psoriasis, rheumatoid arthritis, celiac disease, and multiple sclerosis. So basically, what we're doing here is making sure that the T lymphocytes recognize your own cells as you and does not mistake them for foreign cells and attack them. You do not want your own immune system attacking your own cells. That's where autoimmune diseases come into play. So they have to pass a test to make sure they can do that. T cells that react too strongly to your own self-antigens are either destroyed via apoptosis or made inactive, which is a condition we call anergy. Another thing that can happen is they're converted into regulatory T cells that help keep the immune responses in check. This process is known as negative selection, and it ensures self-tolerance or the prevention of attacks on the body's own tissues. Most T cells are eliminated during positive selection, and only about 2% of T cells survive both tests. The survivors multiply into clones that are specific for particular antigens, forming the naive lymphocyte pool. These naive T cells exit the thymus through the blood and the lymphatic vessels and settle in lymphatic organs like lymph nodes, tonsils, and bone marrow. And there, they just sit there and wait for the opportunity to protect us from disease. Remember, a lot of your cellular fluid with the viruses and bacteria and pathogens are going to make their way to these lymphatic organs.
Bombas makes the most comfortable socks, underwear, and T-shirts.
Warning, Bombas are so absurdly comfortable you may throw out all your other clothes.
Sorry, do we legally have to say that?
No, this is just how I talk, and I really love my Bombas.
They do feel that good. And they do good, too. One item purchased equals one item donated.
To feel good and do good, go to bombas.com and use code AUDIO for 20% off your first purchase. That's BOMBAS.com and use code AUDIO at checkout.
This is the story of the one. As a maintenance supervisor at a manufacturing facility, he knows keeping the line up and running is a top priority. That's why he chooses Grainger. Because when a drive belt gets damaged, Grainger makes it easy to find the exact specs for the replacement product he needs. And next-day delivery helps ensure he'll have everything in place and running like clockwork. Call 1-800-GRAINGER, click Grainger.com, or just stop by. Grainger, for the ones who get it done.
All right, now it's time to talk about B lymphocytes, which we also call B cells. Unlike the T cells, they mature entirely within the red bone marrow. But that's not why they're called B cells. They're actually called B cells because they were discovered in birds in an anatomical structure called the bursa. We produce about 50 million B cells a day, but only 10% survive and enter the circulation. The other 90% are eliminated during positive and negative selection, like we just saw with T cells. B cells that successfully become self-tolerant multiply and form clones of immunocompetent B cells. Those B cells then travel throughout the body. Like T cells, B cells are abundant in lymph nodes, the spleen, bone marrow, and our mucous membranes. Now that brings us to antigen-presenting cells, or APCs. What are these? Well, the reason why we have antigen-presenting cells is because T cells can't recognize foreign antigens on their own, and they require assistance. Not only APCs do this, but dendritic cells, which we saw in the integumentary system, macrophages, and B cells also help out. APCs rely on genes from the major histocompatibility complex located on chromosome 6. These genes code for MHC proteins that have a groove to hold pieces of foreign antigens. With the exception of identical twins, MHC proteins are unique to each person, and they serve as identity markers for your own cells. When an APC encounters a foreign antigen, it engulfs it by endocytosis, digests it, and presents fragments in the grooves of its MHC proteins. We call that process antigen processing. Those fragments are the cell's epitopes. T cells inspect these displays, ignoring self-antigens, but responding aggressively to non-self antigens, triggering the immune response. Communication between lymphocytes and APCs is coordinated by cytokines called interleukins. These are chemical signals that allow immune cells to communicate with each other. All right, so now that we have an idea of what all these different cells are, let's talk about cellular immunity. This is a branch of adaptive immunity where T cells directly attack infected or foreign cells. The immune system remembers these invaders, ensuring faster responses in the future, and there are many types of T cells, but we're only going to really focus on 4 main types. They are cytotoxic T cells, helper T cells, regulatory T cells, and memory T cells. Cytotoxic T cells attack and kill foreign or infected cells. Cytotoxic, it means toxic to a cell. Helper T cells boost cytotoxic T cell activity and assist in both humoral and innate immunity. They assist, they're helpers. Regulatory T cells suppress immune responses once a threat is neutralized, reducing the risk of chronic inflammation and autoimmune diseases. So they regulate the immune response. They're regulatory T cells. Let's make sure that our immune response doesn't overreact. And memory T cells, which are derived from the cytotoxic T cells, remain vigilant against future invasions without causing tissue damage. They remember what we had to respond to in the past, so the next time we can respond more quickly. Cellular immunity unfolds in 3 stages, which are often referred to as the 3 R's of immunity: recognize, react, and remember. So let's talk about recognition first. Recognition involves 2 steps: antigen presentation and T cell activation. So we'll talk about antigen presentation first. APCs that encounter antigens travel to the nearest lymph node and display those antigens. T cells patrol the lymph nodes, checking cells displaying antigens on their MHC proteins. There are 2 types of MHC proteins. MHC1, found on all nucleated cells and present self or abnormal antigens, for example, viruses or cancer cells. And MHC2, which is found only on antigen-presenting cells, and they present only foreign antigens. T cells eventually become limited in their recognition of MHC proteins. This MHC restriction results in cytotoxic T cells only recognizing MHC1 proteins and helper T cells only recognizing MHC2 proteins. So then we get to the T cell activation. Activation starts when a cytotoxic T cell or a helper T cell binds to an MHC protein showing an epitope that it recognizes. For full activation, the T cell must also receive a second signal, which we call co-stimulation. This prevents an accidental attack on the body's own tissues, right? So it's kind of like 2-factor identification when you try to log into a website. Without co-stimulation, the T cell becomes inactive. Again, we call that anergy. With successful co-stimulation, the T cell undergoes clonal selection, dividing to produce effector and memory T cells. The react part is the attack waged against the pathogen. They involve helper T cells and cytotoxic T cells. The helper T cells are the central coordinators of immune responses. When they're activated, they secrete interleukins that attract neutrophils and natural killer cells, and they also attract macrophages, boost their activity, and retain them at the site. Finally, they stimulate T and B cell division and maturation, making more T cells and more B cells. The cytotoxic T cells directly attack infected or foreign cells, like we mentioned earlier. Upon recognizing an antigen MHC1 complex, which is the antigen with the MHC1 bound together, they dock onto the target cell and release a lethal dose of chemicals. Those chemicals could be perforins and granzymes, which we talked about in a previous episode. Remember, they create holes in the enemy cell and then trigger apoptosis. They could be interferons, which we've also discussed before, which inhibit viral replication and recruit macrophages. Or they could be something called tumor necrosis factor, which activates macrophages and kills cancer cells. And finally, remember. Remember is the 3rd R of our 3 R's. Adaptive immunity has a memory. Remember, we've talked about this before. This memory follows the initial response of the cellular immunity. Some T cells are memory cells, memory T cells. They are abundant and have a long lifespan. Their activation is simpler than other T cells and therefore react to antigens more quickly. If you become exposed to a pathogen that memory T cells have been exposed to in the past, a T cell call response occurs. The pathogen is destroyed so quickly that you don't even get sick. In humoral immunity, the body uses an indirect method of defense. Instead of B lymphocytes directly attacking invaders, they produce the antibodies that bind to antigens and mark them for destruction by other components of the immune system. Like cellular immunity, humoral immunity unfolds in 3 stages: recognition, attack, and memory. So now we're going to talk about B cell function. Again, first we have recognition. The first step begins with an immunocompetent B cell which carries thousands of receptors on its surface, each specific for a single antigen. When an antigen binds to multiple B cell receptors simultaneously, linking them together, the B cell internalizes the antigen through receptor-mediated endocytosis. This explains why very small molecules often aren't antigenic. They're too tiny to link multiple receptors on B cells. Once inside the B cell, the antigen is processed, digested into fragments, and key epitopes are attached to the B cell's MHC2 proteins and displayed on its surface. At this point, the B cell itself acts as an antigen-presenting cell. Now, the problem is, by itself, this usually isn't enough to fully activate the B cell. For a full immune response, a helper T cell must recognize and bind to the antigen MHC complex displayed by the B cell. Once the helper T cell binds, it secretes interleukins that fully activate the B cell, triggering clonal selection. The activated B cell rapidly divides by mitosis, producing a battalion of identical cells, all programmed against the same antigen. Some of these cloned B cells differentiate into a different kind of cell called a plasma cell, and it's their job to manufacture and secrete antibodies at a remarkable speed. They can make about 2,000 antibody molecules per second for their entire 4 to 5 day lifespan. These antibodies then travel through the bloodstream and other fluids, ready to neutralize threats. When the body encounters a pathogen for the first time, plasma cells primarily produce IgM antibodies. These are immunoglobulin M antibodies. On subsequent encounters with the same antigen, they mainly produce immunoglobulin G, which provides longer-lasting immunity. Okay, so that is the recognition. Now we have the attack. Once antibodies are released into the system, they use 4 main strategies to neutralize antigens: neutralization, complement fixation, agglutination, and precipitation. Neutralization is basically the antibodies covering critical parts of the antigen. This typically is the regions that allow toxins or viruses to bind to human cells, effectively rendering the antigen harmless. Then, antibodies, mainly immunoglobulin M and G, attach to enemy cells and change shape to reveal complement binding sites. This is our complement fixation. The complement proteins then attach, triggering inflammation, phagocytosis, immune clearance, and direct cell lysis. That's destruction of the cell. This is one of the body's most powerful ways to destroy bacteria and mismatched blood cells. Then we have agglutination. Here, the antibodies cause enemy cells to clump together. Since each antibody can bind to multiple cells at once, they stick pathogens into large clusters. This immobilizes them and makes it easier for neutrophils and macrophages to engulf them. And then finally, precipitation. Similar to agglutination, but involving free antigen molecules rather than whole cells. Antibodies clump soluble antigens into large complexes that can be cleared by the immune system or phagocytized by eosinophils. So that is the attack for B cells. Finally, we have the memory. The first time the body encounters an antigen, it launches a primary response. Antibody production is delayed by 3 to 6 days or so while naive B cells multiply and differentiate. As plasma cells begin secreting antibodies, the antibody titer rises. Now, a titer is the concentration of something in your blood. So the antibody titer is how much antibody you have in your blood. That rising antibody titer consists of these things. Immunoglobulin M, which appears first, it peaks around 10 days and then declines. Immunoglobulin G appears as immunoglobulin M declines, peaks later, and provides longer-term protection. but its levels also eventually fall back down after about a month. However, the immune system does not forget. During clonal selection, some B cells become memory B cells instead of plasma cells. These memory cells reside primarily in the germinal centers of lymph nodes. If the body encounters the same antigen again, memory B cells mount a rapid secondary response, which we call an anamnestic response. Plasma cells start forming within hours, and immunoglobulin G levels surge quickly, often preventing the pathogen from causing noticeable illness. A small spike in immunoglobulin M also occurs but fades fast, while immunoglobulin G remains elevated for weeks to years, offering lasting protection against that pathogen. It's important to note that while humoral memory is powerful, it typically doesn't last as long as memory formed by cellular immunity. Finally, remember that humoral immunity, cellular immunity, and inflammation often work together, forming a three-pronged attack against pathogens.
You buy a pair of socks, that's 2 socks. You buy a pair of Bombas socks, that's 4 socks. Because one purchased is one donated. Socks are the #1 most requested clothing item in homeless shelters. So when you buy a pair of super comfortable Bombas socks, you're also donating a pair. Bombas customers have powered over 150 million donations. So Bombas would like to thank you 150 million times. But we only have like 30 seconds. Go to bombas.com and use code AUDIO for 20% off your first purchase. That's BOMBAS.com and use code AUDIO at checkout.
All right, so that is a lot. That's adaptive immunity in one episode. I know that was a ton to take in. You might want to listen a couple of times, maybe take some notes, because that was a pretty decent amount to process. But now we're done with the immune system, and we're ready to move on to the digestive system in the next unit. So thank you so much for listening. I really appreciate all your lovely comments and emails that are coming through. Please don't forget to follow me on Instagram, and you can check out my YouTube channel. Both of those are Student Help, the number 4, AP. I've been posting a lot of stuff on Instagram lately, just kind of maybe some quizzes, quick little tutorial videos, things like that. So check that out. That's been a lot of fun. And thank you so much, Ryan, for writing from the United Kingdom about the next iteration of Anatomy and Physiology Bit by Bit. I'm really looking forward to coming up with those episodes. But we're going to finish A&P first. And we're going to do that sometime soon, I imagine. But again, sometimes these episodes end up longer than I think, and sometimes these topics take more episodes than I think they're going to take. So I'm playing everything by ear, and I appreciate you sticking with me. Once again, I hope this is helping you get your beer better in A&P. I'll talk to you next time. Anatomy and Physiology Bit by Bit is a production of Minus 55 Media. Don't forget to check out my Instagram account and my YouTube channel, both called Student Help for AP. I'm posting a lot of stuff on there right now that you can watch and that will hopefully really help you understand this A&P stuff. Very special thank you out to my family, Bucks County Community College, and McGraw-Hill Education, where you can find my tutor video-based textbook alternative Anatomy and Physiology digital suite, already being used by several colleges and universities throughout the United States.